This is episode 53 of the Immunology Podcast, Immunology of Infectious Diseases with Dr. Prasanna Jagannathan. Hey everyone, this is Dr. Jason Goldsmith and Dr. Brenda Roud. Welcome back to the Immunology Podcast, where we have conversations with immunologists. The Immunology Podcast is brought to you by STEMCELL Technologies, a global biotechnology company supporting life science research and fostering communication and collaboration in science. If you enjoy the Immunology Podcast, please rate us and leave us a review. We're always looking for feedback on how the podcast can be improved and for suggestions on guests. Today, we have Dr. Prasanna Jagannathan from Stanford University on the podcast to talk about his research on the human immunology of infectious diseases. We've also got our usual roundup of recent highlights in immunology news coming up. But first,
only 2 days until Immunology 2023, the annual meeting of the American Association of Immunologists. Jason and I and the rest of the Immunology Podcast team will be attending the meeting, and we can't wait. Make sure to stop by the Immunology Podcast booth in the exhibit hall to win some prizes and learn how your research could be featured in an upcoming episode. Learn more at www.immunology2023.org. And please, let's see you there.
hey, so I get to finally meet you in person after our last failed attempt.
I know. I'm so excited, Jane. I'm so excited. It's going to be a lot of fun. I'm definitely looking forward. And it's a jam-packed session. Have you been over the program?
No, I'm going day by day here. So I fly out to Digestive Disease Week ahead of this recording. And then from there, I have a day in DC to recover and have work meetings. And then I'm going to pick out all the things in the program I'm going to do. And then I'm going to be at Immunology 23. And then after that, I'm going to a microbiome summit in Rutgers. So my family is not real thrilled with me right now.
No, someone's going to be away. It's like a sailor. He leaves for weeks at a time. Well, but it's going to be fun. I've been looking at the program and I have to say it's hard to choose because there's so many parallel sessions. I'm getting a little bit of analysis paralysis right now.
Well, the good news is there's two of us.
True. We need to divide and conquer, don't we?
But I would be remiss to also mention other good news is the drug I've been working on for the last 2 years was approved.
Congratulations. So what's exactly the drug? How can we go to the pharmacy and buy it? Is it like a book?
So it's specialty pharmacy only. It's called VOWST, and it's for prevention of C. difficile infection. So after you get it, to prevent you from getting it again.
And it's made of spores, right?
Made of bacterial spores. Yep.
Very nice. Congratulations, dear listeners. You don't know because you're not in the behind the scenes, but I have been hearing about the progress of these particular item for months. And, it's a lot of work here from looking at it from the other side and just trying to support here my good friend. But man, Jason, that's a lot of work, wasn't it?
It was a hairy few months, but now there's all smiles.
Well, congratulations. I hope you got a nice bonus for that.
I hope so too, but relax, get to go to Immunology 23, hang out in DC with some awesome people. It'll be a good time.
You can be a good resource if any of our listeners is also interested in pushing a drug through FDA approval. You have it all fresh in your mind, don't you?
That is very true. Talk about all those biopharma experiences. I guess we got to start talking about drugs and papers and stuff. So I'll start with a drug. So this one is methotrexate suppresses psoriatic skin inflammation by inhibiting neuropeptide transporter SLC46A2 activity. I'm sorry, folks, if I cough a little bit. I'm getting over a really bad case of laryngitis and had to bite the bullet on antibiotics recently. So it's an immunity, came out April 27th. First author is Ravi Bahadarwaj, and last author is Neil Silverman. So high level, I'm gonna go into intracellular pattern recognition receptors. So the NOD family, right? You got NOD1 and you got NOD2. And just for those who reminder, if you get one of these neuropeptides or peptides that can get intracellularly, pulled in by a cell, so kind of like toll-like receptors, they also involve MyD88, but these are intracellular receptors. You've got NOD1 and NOD2. NOD1 is typically, we use DAP to activate it. And just a little bit of background, that can induce cell damage and death and inflammation, everything. So they show a couple things. I'm going to go a little out of order, but I think it's important just to give the story. So they show in epidermal keratinocytes in that system with psoriatic-like component, NOD1 activation, where you put bacteria on it that can cause psoriasis, or imiquimod, which is an immune activator, can cause psoriasis. You get pyroptosis through caspase-1 and our favorite gasdermin D. Who doesn't love, to call it back to gasdermin? So
all the roads go back to gasdermin, don't they?
They do.
Go on. Well, sorry.
Yeah, exactly. It keeps going. But how does it get in? So there is this family called the SL. I'm going to try to get the full name of it because it's a zesty name. The SLC family, but they're known as solute carrier 15. So SLC 15 family, but in this case, there is a new family called SLC 46. I don't know why it's 46. Please don't ask me. So they demonstrate, and it's been known that there's been some NOD-dependent NF-kappa B reporter activity with these, but they demonstrate that DAP gets in through SLC46A2, and NOD2 ligands get in through SLC46A3. So you can use IE DAP or MDP as to stimulate. If you knock out either receptor, either you lose NOD1 or NOD2. So 46 goes with A2, the smaller number goes with NOD1, and 46A3, the bigger number, goes with NOD2. That's how I remembered it. Smaller went with smaller, bigger went with bigger, because sometimes they have to be simple that way. And so they do knockout mice. They demonstrate that a NOD knockout and an SLC knockout phenocopy each other as appropriate. So that's how the ligand's getting into signal NOD. So that's really important to know. Then you can target the receptor. Well, there have been some data that methotrexate and other things affect these pathways. And lo and behold, methotrexate is an inhibitor for SLC46A2 and to a lesser extent A3. And they show this with a bunch of immunofluorescence experiments showing that this, marked chemical can get in. If you put methotrexate, it does not get into the cell. And so long and short, now you're wondering, well, topical methotrexate, so not oral, which has its own issues, right? But topical methotrexate could be a treatment for psoriasis by blocking the microbial driver of it, microbiome, because why not add microbiome in if we're talking about NOD and gasdermin, or getting all of my favorite flavors of ice cream in one paper sundae here. We can get this microbial driver of signaling blocked off through a methotrexate as an inhibitor of the receptor. So there you go. And, I like the paper. I think it gets some good signaling. It gets some good druggability and all of that.
So basically everything you like in science in one paper. How happy are you about it?
Very happy. I mean, if it had Fret, I'd love it even more, but it's okay. I can't get everything all in one.
Yeah, I guess you get what you can. But I'm always interested when you see these very old drugs, like methotrexate has been used for cancer. I mean, I don't know. Is it used? I don't think it's used anymore very much.
It's not used for cancer a lot. It's used in inflammatory bowel disease as an adjunct immunosuppressant.
Right. But the way it acts is completely different.
So it's used for women's health occasionally.
But then it works through a completely different mechanism, right? So for what people thought methotrexate did in cancer cells than what is doing here.
Yeah, it's just a straight cellular transport inhibitor. So it has a completely other mechanism, which really makes you wonder, like, how often we get the mechanism right.
Right. That's...
like, how bad are we at that?
Yeah.
Probably pretty bad.
Yeah. Well, it worked for a while. And it's nice to see that you can repurpose some of these drugs. Maybe, you can use for something better. All right. I'm going to talk about our next paper, which also has the things I like, which is regulatory T cells, great cells, awesome people, awesome cells in here to have in your body. So Tregs in tumors, bad news. First, let me go into the authors and then give your credit into the authors. So this paper is called Systematic Elucidation and Pharmacological Targeting of Tumor-Infiltrating Regulatory T-Cell Master Regulators. It was published in Cancer Cell on May 8th. Alexander Obradovic, Casey Ager, and Mikko Turunen are first authors from the lab of Andrea Califano at Columbia University. So what did they do? They looked into tumor infiltrating regulatory T cells into different in samples from different types of tumors from human patients. And they did this analysis in which and I think this is very important, they compared the RNA-seq, so the transcriptome of Treg. So they sorted Treg populations from tumors, and they compared them with other T cells in the tumor and with peripheral regulatory T cells from the same patient. So they took the transcriptome and they used a couple of algorithms, which are, I don't completely understand the fine grain, but they used two algorithms, one called Arachne and one called Viper, in which they took the transcriptomic signatures of these cells and they inferred regulatory networks. So basically, which genes are regulating this particular transcriptome in order to identify what they call master regulators, which would be a transcription factor or a particular receptor that initiates a particular transcriptomic program. And that's how kind of they evaluate, and by comparing the transcriptome through this algorithm of different types of cells, they identified what they call master regulators that characterize regulatory T cells in the tumor and are different from either regulatory T cells outside of the tumor, the ones that you need for homeostasis, basically, and other T cells inside the tumor that are not regulatory T cells. For example, CD8 cells that you want to keep going in the context of a tumor response. So doing this, they use, machine learning, random forest algorithms. They use some classificators. So basically, they use a lot of bioinformatics to come up with 17 different master regulators that seem to be driving the particular phenotype transcriptome and therefore the phenotype of the regulatory T cells in the tumor. And basically, they tested these ones in vitro, and they show that if, for example, if they overexpress some of these master regulators, they can induce the signature in cells in vitro. And then they ended up identifying also through a knockout experiment in mouse models. They also identified that if they knocked out, they depleted some of these candidates, they would also deplete regulatory T cells in the tumor of the mice. So they use a mouse model called CHIME, which basically is I think is very interesting because they basically reconstitute a mouse that is with a bone marrow that's expressing Cas9, and they transduce the bone marrow with a library of different guide RNases, in this case against this different master regulator candidates, and they show that then there's a depletion of certain particular master regulators in a Treg that they see in the tumor. And they come up with that one. There's a couple of top hits here, but I think the one they focus on the most is an enzyme called TRPS1. And TRPS1 it really comes up in their analyses, and they show that by this particular gene is a transcriptional repressor, and which didn't really have a previously known function in T cells. So their top candidate from this analysis is a gene called TRPS1, which is a transcriptional repressor that didn't really have a known function in T cells. So they were very happy that they found something that was truly unknown. And they kind of show that by depleting TRPS1, it does really negatively affect the Tregs in the tumor infiltrating regulatory T cells, while it doesn't affect either peripheral Tregs or other T cells in the tumor. And I like this about the paper. I think it's very important to look into the kind of how transcription factors go beyond the transcriptome and to trying to understand like the dynamics that this transcriptome brings. But then this paper has a second part. And here I find I have a little bit of an issue with it because it feels like two separate stories that the only thing they have in common is that they're looking into regulatory T cells in the tumors. So they find this compound called gemcitabine, which is used for cancer chemotherapy. And they also show that by using low dose of gemcitabine that is below what is used for chemotherapy, this actually seems to target specifically regulatory T cells in the tumor. And together with anti-PD-1 treatment, it really induces tumor rejection in their MC38 mouse model. They also suggest that this is through, to some extent, is targeting the regulatory network that stems from TRPS1, but I'm less convinced of that from their data. So that's why I feel like sometimes they don't feel very connected, these two stories. But I think in any case is a very interesting result that they have. So master regulators from using these fancy algorithms from their transcriptomic data, and a new compound that we already used, like methotrexate, we're repurposing an old friend.
Yeah, I think that it's interesting that it so specifically targets a subpopulation. I wonder, there's got to be something like pharmacokinetic or dynamic going on. It's like preferentially making them go in. Do they ever solve why it's so exquisitely just that subpopulation? Because that's weird to me.
Well, I guess that we know that Tregs in the tumor, or like they're quite activated, and we know that they have certain markers, or they have certain proteins that are expressed that are different to the ones in the periphery. Often Tregs in the periphery have a less activated phenotype. So I am not that surprised myself. There have been other proteins. I think a good example is CCR8 that has been associated with Tregs in the tumor. It doesn't seem to be necessary, but they do seem to express this chemokine receptor to levels that suggest that depleting CCR8 might actually help reduce Tregs burden in tumor. So I'm not that surprised. They're just, but it wasn't easy to find. But I'm not surprised they exist, this regulators.
Interesting. All right. Well, to continue discussing regulation, I'm going to talk about dynamic chromatin accessibility licenses STAT5 and STAT6 dependent innate light function of Th9 cells to promote allergic inflammation. Nature Immunology, April 27th. First author is Aaron Sun. Last author is Daniela Schwartz. So this paper has a lot of detail. I'm going to try to distill the high level. So IL-9 is a cytokine produced by Th9 cells as a major driver of allergy responses and in multiple systems. And so they use some of those models here. But what's weird about it is that Th9 cells aren't stable. You can barely generate them in vivo. And like once they start making or IL-9, they drop off over time. And one caveat for this whole paper is mice are more unstable than human for all of these studies, but they find the same thing. And so they do an exploration and really they determined that it's actually bystander activation. So they purified the cells, they get them to produce what they want. They withdraw T cell receptor stimulation, but find that the Th1 and Th2 in this population stop doing their thing without TCR, but the Th9 keep going and don't lose their re-stimulation even. And so they don't degrade like you usually do when you make a monoculture. So if you put Th1 and Th2 cells with Th9 cells, the Th9 cells keep going. They went, well, this looks like there's some bystander activation going on. And so what they found is that, in fact, it is paracrine cytokines, IL-1, IL-2, that can drive Th9 to keep going. And then they go down further and find that, so it's very specific bystander effects. It's not NF-kappa B driven or MAP3K, very little effect there. They have some, but they find it's STAT5 and STAT6 mediated. And if you use a STAT inhibitor, a pan-JAK inhibitor to block STAT signaling, you lose this bystander effect. So at a very high level, and we've known that JAK inhibitors could infect allergy, but it's such a big gun, they didn't know how, right? And so what this paper really does is it establishes that these Th9 cells, and have a STAT5 and 6 program, and they actually look at chromatin remodeling, they look at accessibility of the chromatin and see accessibility naturally degrades over time. But if you hit it with these bystander cytokines, the STAT5 and 6 signaling will keep the chromatin open and enable it for longer, although eventually they do exhaust and turn off. And so you have this bystander effect that drives these Th9 cells to keep going. So moral of the story, allergic reaction and it's independent TCR. So they can ablate TCR function using tamoxifen-induced system or blockade. You don't need the TCR once it exists. You got to get the cell to exist. But once you have some Th9 producing, Th cells going on here, if you can get rid of the TCR and just have bystander IL-1, IL-2 driving STAT5 and 6, and it keeps these going. And that's why they're also unstable, is without this secondary bystander effect, the signaling goes away. So it's kind of that self-regulation as well. It depends on the paracrine stimulation to maintain its strength. So there you go. Networked immunology.
So I wasn't completely, so this is in the context of allergy?
In context of allergy.
So does this apply for humans? Do we see Th9 in their responses? Is that something that they observe in actual humans?
Yes, and in human cells that they take out and stuff, but yes. So this is known in people that Th9 is important. There's just how do you abrogate it and what's going on with these cells, and understanding the biology of these T cells has been hard because they try to stop. And what they see is this chromatin remodeling suppresses the activity over time, even in the presence of the bystander cytokines.
Okay, so another leg to the allergic response.
Yep.
Yet again. Okay. All right. Then to finish our science jamboree today, second paper of the day. It's actually, I'm going to cheat a little bit because I'm going to talk about one paper, but I'm going to make an honorable mention to another paper which came out almost at the same time, and they're both looking at the very similar new technique, which is very close to my heart because this is a new way of genetically editing cells. With they're focusing on T cells, so also that's good. And basically, this might be a revolution in how we do CRISPR, because in this case, usually what the most difficult part of doing CRISPR, especially if you have like primary cells, is getting the CRISPR to the nucleus and getting the Cas9 to the place it has to act. And at least in the context of immunology, the most common way of doing this, or a very popular way of doing it, by electroporating, for example, ribonucleoproteins. It is a Cas9 loaded with a guide RNA. You electroporate cells, for example, T cells, and then you get the RNP, the Cas9, inside the cell. And then these RNPs are engineered with nuclear localization signals that will send it to the nucleus where it has to act. But for those like me who do this, there's a huge limitation in the sense that electroporation is not super nice for cells. And often you lose a lot of your cells that don't survive after being zapped by hundreds of volts. And that reduces the effectiveness of what you're studying. You don't get that many cells. And for the purpose of making therapeutic cells, usually the rule of thumb is that the more cells, the better. So that's why I was so interested when this paper came out. This is called peptide-mediated delivery of CRISPR enzymes for the efficient editing of primary human lymphocytes. That's published in Nature Biomedical Engineering. First authors Dana Ross, Joseph Muldoon, and David Nguyen, from the lab for a collaboration of the labs of Ross Wilson, Justin
Aquem and Alexander Marson. They're all the different types of different universities of California, Berkeley, San Francisco. And they've teamed up to bring a new way of getting your RNP inside your cell. Basically, you combine it with a peptide, and this is it's so easy, it's insane. They combine it with a peptide that has the ability of kind of making its way through the cell membrane. So, and that gets the RNP inside the cell. So, what they do is, we know that we have they exist what I call cell penetrating peptides that are common from viruses uses peptides to go through the cell membrane, and they have been used for other purposes, for other kind of payloads. So we know they exist, and there's different versions. You can fuse them from different types of peptides, and there's one particular types which are chimeric peptides that contain fusions of what is known as an HA2 derived endosomalytic peptide and a cell-penetrating peptide. For example, one known as TAD that is derived from HIV, and the endosomalytic peptides like HA2 from influenza. And these are important because they disrupt the endosomal membrane and they facilitate the translocation from a vesicle that is taken up into the cytosol. So they make a different variants. So they know that these guys can help get things through the membrane. So what they start doing, they make a lot of different variants, and they end up finding a particular combination of amphiphilic peptides that allow the RNPs to get into the cell. So they test 37 different variants, and basically they found one characterized by a sequence A5K sequence that really does the trick and allows you to get the RNP inside the T cell without having to electroporate or use a virus for that matter. And that seems kind of something very simple, but it might be quite revolutionary because then that means you don't need to get an electroporator in your lab. You can just basically incubate the cells with your RNPs with these peptides, and then it works. So it works fairly well for knockouts. They do have less efficiency than if you electroporate the cells, but they make it up with lower toxicity. They show that they can do sequential treatment, so they can knock out different genes in sequence. So they do one, then a couple of days later, they do one other round with a different gene. And this prevents having different double-strand breaks at the same time, which might result in translocations or weird things happen with the genome. So they reduce that risk by doing it sequentially, which you cannot really do with electroporation because the cells don't survive. And they show that this works for B cells, T cells, and K cells with different rates of success. They even show that you can combine it with a viral vector, such as adeno-associated viral vector, to make a knocking. Again, in general, the efficiencies are lower than if you do it with electroporation, but you get more cells. So, depending on what you're looking for, this might be the thing for you. And I think what's also very important is that they do some phenotypic analysis and suggest that really the perturbation of the phenotype of the cell is minimal compared to what electroporation does to cells when it comes to extracellular markers. And they do a bunch of analyses and really suggest that this is much more gentle on the cell. So very interesting. And then I just want to shout out to another paper that was also published. This was in Nature Biotechnology the day before. This one actually, I should discuss this one more, but this is first also Zhen Zhang from the corresponding authors are Junwei Shi, Shelley Berger, and John Wary at University of Pennsylvania. And basically they reach a very similar conclusion. They start with a slightly different approach because they don't use regular Cas9. So I think also what's very important of the previous paper is that they just use Cas9 protein that is available commercially. Nothing special. You just add the Cas9, and on top of that, a peptide that you can buy. These guys, they start already made a modified version of the Cas9 in which they add a TAT sequence to it to make it kind of already self penetrating the RNP itself. And they show that on top of that, they need to add a peptide that contains a TAT sequence as well to improve the efficiency of the whole system. But in general, they also reach the conclusion that by using these peptides, you can really kind of even replace the need of having to electroporate the cells. They don't do, like the previous paper, like a side-by-side comparison with electroporation, which I would say is a standard in the field at the moment. But they do make a really strong argument that this also works with a slightly different peptides, slightly different conditions, but the idea is the same. So very interesting new development in the field. We'll see how other labs are going to start replicating. I'm pretty sure many are going to jump into the no electroporation wagon, and we'll see how that goes.
So do they have to covalently link the peptides, or they just get surrounded by the peptides and they serve as a carrier?
Yeah, so in the case of the first of the paper from University of California, they just do need to co-culture, and they seem to form some kind of complex because when they measure the size of the particles become bigger. So the RNPs are clearly kind of surrounded by these peptides. In the case of the construct from University of Pennsylvania, they actually have a version of Cas9 that has a fusion with a TAT on top of the nuclear localization signal that all of them have, and then on top of that, they add an extra peptide to kind of help further.
Awesome. All right. Well, we're going to see each other very soon here. And in addition, we're going to be speaking with Dr. Prasanna Jagannathan at Stanford University in just a moment. But before we get to that, we'd like to remind our listeners about Immunology of Infectious Disease News, a free weekly newsletter brought to you by STEMCELL Science News. It summarizes all the latest research, news, jobs, and events in infectious disease. And Immunology of Infectious Disease News helps you stay current with the latest COVID-19, HIV, hepatitis, tuberculosis, influenza, and malaria research, which we discuss here soon. Subscribe at immunologyofinfectiousdiseasenews.com.
Welcome, everyone, to this week's episode. We are joined by Dr. Prasanna Jagannathan. He is an assistant professor of infectious diseases and microbiology and immunology at Stanford University. And he's going to talk to us about his research on malaria, also maybe on COVID, and a little bit about his very interesting career path that I hope that we can discuss today. Thank you so much for joining the Immunology Podcast.
Thank you, Brenda. Thank you, Jason.
Thank you for being here.
First, diving into your research, I am very interested in, and also I think malaria is such an important research topic. But I wanted to ask you something. I noticed from your CV, from your experience, that you started your studying religious studies at the University of California, and then you went on to medical school. Maybe I am not the only one to think that's quite a curious career path. And I was wondering, maybe you can share with our listeners, how did that transition come to happen?
Yeah, I mean, sure. I'll try to be brief, but I think one of the amazing things about the American educational system is that it allows you to take detours in your education and take some unusual kind of career trajectories. And so, I started at the University of California, Berkeley as an undergraduate. And when I started, I initially thought I was interested in science, but I was very, when I went into my first chemistry class of 800 students, and I was sitting in the very far back, it was very intimidating. And I found it very difficult actually to kind of engage with science as an undergraduate. And during my first year at Berkeley, I took a religious studies class. It was a religious studies upper division class in South Asian religion. And for me, as an immigrant, as a child of immigrants, it was kind of a beautiful introduction to a college life and exploration of identity. And I just fell in love. I fell in love with the critical kind of thinking aspects of thinking about not just kind of my religion, but kind of religions of South and Southeast Asia. Thinking about the kind of political economy of religions, and it was really something that happened kind of very early on when I was an undergrad is just kind of exploring that identity myself, as well as a space that allowed me to really think critically. So I spent during my junior year, I actually lived in India for a year, and I studied South Asian religions. During that period of time, I actually got the opportunity right next to where I was living. There was one of Mother Teresa's homes for the dying and destitute, and so I got to volunteer there as an undergraduate. And for me, that kind of was the first time in my life where I realized, that I really wanted to do. I really wanted to spend my life working in an applied way, and to address, especially social inequalities that are important in our times. And so when I came back, actually after that year, I refocused. Actually, before I went to medical school, I actually taught high school. I was a high school science teacher. I did it through Teach for America, and taught high school science in Southern California and Los Angeles. Spent a few years doing that. That was actually the hardest thing I've ever done in my life. That was the most challenging profession. I have an incredible respect for teachers through those experiences. Like through that, realized that I could potentially make a big impact through healthcare. And so I actually decided to go to medical school and went to medical school after that.
I'm so glad I asked. That's such a wonderful career arc. It's fascinating. It's really, really interesting.
So having gone through med school myself, I know sometimes there's defining moments in your life or a defining story or something that makes you go, either that's what got you to med school, or you keep remembering while you're in med school when you would rather quit and open a coffee shop at certain points. From your travels and your experience teaching, is there any one such thing that stuck with you kind of as that beacon when you went to med school then on?
Yeah, I mean, there's two things. One, I always think back to this, so I was a religious studies major in undergrad. I really never held a pipette. I wasn't really, I never worked in a lab as an undergrad. And I went to medical school, and I actually went to medical school and had the distinct privilege of being able to work with Partners in Health. And Paul Farmer, who passed away last year, but I was really fortunate to have those opportunities to work with that organization and think about kind of the social determinants of health, and think about kind of our role as providers of health in kind of addressing social inequality and social injustice. Specifically, I was interested in kind of the nexus between social justice and infectious disease, because there's such an important relationship there. And but I remember early in medical school listening to a lecture by Bruce Walker. I don't know if you know Bruce Walker, but he is a professor at the Reagan Institute at Harvard, and just a wonderful physician scientist who's spent his career studying the human immunology of HIV infection. And he gave just a very compelling talk when I was, I think, a first-year medical student. And for me, that was one of my first times thinking about the potential of the human immune system. And so I decided actually to take a very different trajectory in medical school. I actually took a year off in med school, and I joined the Howard Hughes Medical Institute NIH Research Scholars Program that was actually run we were housed in the cloister on the campus of NIH. And I spent 18 months there in the lab of Mark Connors and Steve McGillis, who are also kind of human immunologists, and think about CD8-mediated control of HIV infection. And for me, that was my first experience holding a pipette. It was my first experience asking a hypothesis, a true hypothesis. It was my first experience failing in a research experiment. It was my first experience of having, of being successful in an experiment as well. And that for me was, just completely career altering. I just fell in love with research after that. And I fell in love with the idea of asking questions. I fell in love with the potential of the human immune system. And for me, with my winding kind of path through, religion and social justice, it again took yet another turn, and I became fascinated with, how we could potentially harness the immune system to address really important problems that face us.
I guess that probably your current focus in your lab of malaria and your collaborations, you have a lot of collaborations with people in the ground in Uganda in particular, if I understand correctly. Probably is also related to that original interest on how medicine can advance many people at the same time. Can you tell us a little bit about what your lab, now we can ask you more about your science and your research. What is your lab focusing in when studying malaria? And also maybe you can tell us a little bit about how did it come about studying COVID as well as part of your research?
Sure. Yeah. So when I was in my clinical training, I did my clinical training in internal medicine and infectious disease at UCSF. And during my clinical training, I had the opportunity to go to Uganda. Well, actually, one of my residency attendings. So these are the people that help us learn about how to be doctors. His name is Grant Dorsey, and at the time he said, "Hey, we're doing this clinical trial in Uganda. You should come and work with us in Uganda and learn a little bit about malaria." And for me, like that, I had really very little. I knew very little about malaria at the time. This was, as a clinical trainee. And so I spent a few months in Uganda, and that's, when I went there, and learned about this disease, how impactful and important it is in places like East Africa, in Uganda, for example, where we've worked, and also like how fascinating a disease it is. It's a disease that we've essentially evolved with. I became really excited about the possibilities of working in the context of malaria and asking important questions about naturally acquired immunity to malaria. And in places where we work, for example, in Eastern Uganda, kids get infected over and over with malaria. So when they're young, when they're less than 5 years of age, for example, some kids, on average, kids are getting malaria 4, 5, 6 times a year. Just a ton of malaria, and every time they get malaria, they're at risk of dying. They're at risk. They're sick. Their parents or their guardians have to bring them into the clinic to get them treated. They lose work. It's an incredibly impactful disease. It's really unlike any disease that we deal with, for example, in North America and Europe, for example. And so the societal impact is really profound. But what's also fascinating about malaria is that it's a disease that, with repeated exposure, children are eventually able to tolerate parasites without developing symptoms. And this is uniform, such that, like, if you go to school-age children, for example, and you test them for the presence of malaria parasites in places where malaria is highly endemic, almost all school-age children will have evidence of malaria parasites, but they just won't have symptoms. Similarly with adults, they'll have lower levels of parasites that might not be detected by microscopy, that need to be detected by PCR, for example, but you can detect parasites in almost everybody. And so it's this disease where when you're very young, you get sick over and over again. But if you survive those early life challenges, then as you get older, you're able to tolerate parasites without symptoms. And so this was fascinating to me. This is fascinating to us as malaria immunologists, thinking about the mechanisms by which the immune system actually evolves this capacity to tolerate parasites without symptoms. And so that's the main thrust of our lab, is really trying to understand, what are mechanisms that actually facilitate the development of asymptomatic parasitemia, and then the other question that we have as a lab is, how does intervening, for example, with highly effective preventive interventions like chemoprevention, for example, how does intervening early in life impact this acquisition of naturally acquired immunity or exposure-dependent immunity? And so those are kind of the two series of questions that our lab has really been focused in over the last several years.
And I wanted to hone in on that bit now a little bit. You had mentioned, with this focus, how early exposure changes things. And if I was reading your research right, and correct me if I'm wrong or elaborate if I'm on the right path, part of it matters that it's a kid being exposed, right? If you were just to hot drop an adult in at age 30 and expose them to malaria a bunch, they're not going to get the same pseudo tolerance to in the same way that you get if you started with as a kid. So if that's correct, or if I'm not, put me on the right path, what is it about that childhood immune system that's different, that's enabling this?
It's a good question. There actually are not great examples of experiments where we actually take adults and repeatedly expose them. There were forced experiments in the 1940s and 50s of prisoners, to treat syphilis, and so we actually experimentally infected prisoners. And there's some data from the 1940s and 50s that over the course of repeated infections, the fever threshold, or the temperature that was experienced by these individuals actually declined with repeated exposure. There's actually now some controlled human challenge experiments that are actually testing this. In Edinburgh, for example, they're actually testing in adults what happens when you repeatedly infect an adult, and are finding very similar things, that with repeated infections, adults also eventually acquire this capacity to have asymptomatic parasitemia. And so I do absolutely agree with you that there's something that's somewhat unique about the developing child immune system, or the infant immune system. These infants are exposed early in life. They're exposed to parasite antigens in utero as well. So this exposure kind of occurs very early in their lives. But this process of kind of exposure-dependent immunity, I think, also occurs in adults as well.
Can I ask a very basic question regarding how malaria and the disease it causes really works? So if you have as asymptomatic parasitemia, how bad is that? Because I always understand that the worst part of malaria is the fever and the pain that you get from the immune reaction against it when the parasites are in your blood. But then what is the advantage, and how did we evolve to, or how does it make sense to evolve to have this maybe tolerance? Is that a correct word?
Yeah. I mean, I think infection tolerance or disease tolerance, it's distinct from kind of our immunologic tolerance, although there's probably a little bit of overlap there mechanistically. I think we evolved with it because it's a way that allows for mutual existence of both the parasite and the human. And on an individual level, being able to tolerate the parasite without having symptoms or sequelae is obviously advantageous to us as humans. But it's not all like rosy. If you have asymptomatic parasitemia, there's actually a fair amount of evidence that asymptomatic parasitemia in school-aged children is associated with worse cognitive performance and worse school performance, for example. The most important thing is that if you have asymptomatic parasitemia, you're still a transmitting host. So if a mosquito bites you, for example, and then transmits it to a pregnant woman or a young infant, it puts them at risk. And so it really kind of establishes it maintains a reservoir of ongoing transmission that I think at a population level is really what we want to what we want to eliminate. So I think on an individual level, there's benefits because it allows us to survive, but on a population level, and also even at an individual level with regards to kind of unanticipated consequences of parasitemia, I think that there's real issues with it.
All right, so diving head in now, malaria. You've kind of described how it's a scourge. I think a lot of people have heard about how much of a scourge it is. What do you think are the promising treatments out there coming up for it?
Yeah, no, it's a great question. I mean, I think one thing to remember is that we have highly effective curative treatments for malaria. There is a specter of drug resistance that's there. It's a very real specter, but 2023, if you are a child living in African settings, you have access to treatments that are 100% curative. The issue, though, is that these curative treatments don't prevent you from getting reinfected. And so the most commonly used antimalarial artemether lumefantrine, if you're infected with malaria parasites and you're symptomatic, you get this treatment over 3 days. The parasites will go away and you'll be fine, but then you'll be at risk for reinfection within a couple of weeks. and so these kids will get reinfected again. So when I think about really promising interventions, we actually are on the cusp, I think, of some incredibly important breakthroughs, and that is really to try and prevent infections from the first place, and or potentially engender a better immune response against the parasite to prevent infections. And so I think there's two kind of classes. One is the vaccines, and so the malaria vaccine that's been approved was approved at November 2021, I think. Was the RTSS vaccine. That vaccine was in development for decades, or has been in development for decades. The first phase 3 clinical trials were conducted more than 10 years ago, or were reported more than 10 years ago, and they showed that at least in the first year after a vaccination, children were protected about, it actually had a protective efficacy of around 50%. So not fantastic, but certainly better than any other vaccine candidate that had been tested before then. The vaccine efficacy wanes actually fairly rapidly, such that over a 3 to 5 year period, the efficacy is probably closer to 30%, which again isn't great. But if you think about this idea that kids are getting repeatedly infected, even averting 30% of cases, for example, represents a really big win, I think, for the malaria prevention community. So that's one vaccine that was approved by the WHO and is slowly starting to be rolled out. There's issues with production. GSK is not going to provide this vaccine at large scale, but they are committed to providing it in some smaller scales, and then they're also committed to licensing this technology to a company in India, Bharat Biotech, I think, that's going to produce this vaccine. So that's one exciting development. The other exciting development is kind of a second generation or next generation malaria vaccine called R21 that was developed at Oxford. So this vaccine is very similar in ways to RTSS. It's the same antigen. It just has a slightly different ratio of the CSP molecule, which is the vaccine antigen in the nanoparticle in relation to hepatitis B core antigen. And so this R21 vaccine, there's been published results from phase 2 trials that show it to be very effective, about 60 to 70% efficacious in the first year after vaccination. And the phase 3 trial results haven't yet been published, but they were presented a few months ago at a meeting that suggests very high efficacy in multiple settings in phase 3 trials as well. So this vaccine has an advantage. It's in the end, it's going to likely be very similar to RTSS, but it has the advantage that it's going to be produced by the Serum Institute of India, and it's going to be potentially much cheaper than RTSS to produce, at least. Well, at least that's the hope. And so there very well could be a bigger market for R21 to be out there. So I think vaccines is one big aspect that we're all very excited about in the malaria kind of community. And then the other kind of more interesting therapeutic or preventative intervention that I think we're very excited about is this idea is our monoclonal antibodies. And so the NIH, in the Vaccine Research Center, Bob Cedars lab, developed several years ago a monoclonal antibody, CIS43LS, that they tested in phase 1 clinical trials, found to be very effective in phase 1. And late last year, the NIH Peter Crompton's group tested this vaccine in Mali and found it to be highly protective against infection in Malian adults. And so this was the first generation monoclonal antibody that was given intravenously. They have a second generation monoclonal antibody, L9LS, that can be given subcutaneously, and that monoclonal antibody is being tested in younger children, both in Kenya and in Mali. And there's a lot of momentum to try to make it's so effective that there's a lot of momentum to try to make this more widely available and to actually potentially produce it at scale at a cost that's reasonable. I think there's still a lot of logistic hurdles to kind of make that happen, but I think there's a lot of excitement and commitment within the research communities to kind of to translate this highly protective intervention to the field.
That is super interesting. I was not aware that the monoclonal antibodies were so advanced They were so promising. I remember discussing the results of the RTSS vaccine trial during a vaccinology course at university. So that's how long it's been since that vaccine has been on development, clearly. But it's good to know that now there's better alternatives. New therapies are being delivered in a way. Always malaria is such a understudied, or given the magnitude of the impact that it has on human life, it always feels like it's a little bit understudied.
To get to the immunology of this a bit, why is it so hard to gain an immune response to malaria? Like, you get infected, you get treated, you get infected 2 weeks later. That's pretty bad.
Yeah. I think there's a couple of different aspects that make our immune system pretty ineffective at kind of dealing with repeated infections. One is that it's a big organism, more than 5,000 genes, lots of antigenic variation. The organism actually has, this capacity to not just have diversity, but also it can epigenetically modify the antigens that are actually on the surface of an infected red blood cell, for example, to evade the host immune response. And it's quite effective at doing that. And I think it's some think of it as kind of as a battle kind of back and forth between the host immune response to kind of get at variants and get at these antigenic targets. I think that is very important. I think that there's other things as well. Like, one aspect that our lab has really been interested in is this idea that malaria itself induces an incredible inflammatory response. There's lots of innate immune sensors that can recognize the parasite that actually produce a pretty profound inflammatory response that are likely responsible for the symptoms of infection. And that inflammatory response pretty rapidly engenders a regulatory response as well to really dampen down that inflammation. And the impact of that regulatory response on susceptibility to reinfection, for example, I think is very important. I think a lot of us have been thinking about the role of regulatory T cells, for example, not conventional FOXP3 positive Tregs, but other non-conventional regulatory T cell populations like type 1 regulatory T cells, and thinking about if those are induced, for example, following a malaria infection to kind of help deal with inflammation, what's the impact in terms of both on antigen persistence as well as the risk of reinfection. That I think is also likely plays an important role in our inability to kind of deal with reinfection following repeated infection.
Also, if I may ask about one of your recent publications, you also looked at the role of members of the innate immune system, particularly NK cells. What can you tell us about their role? And I guess, particularly in young children, they might even be more relevant to the protection.
So, I think what's fascinating, I think, about our understanding of immunology over the last 10 years is that our appreciation that there could be adaptation within the innate immune compartment has really grown. Like, we understand now that innate immune cells, myeloid cells, hematopoietic progenitor cells, natural killer cells, gamma delta T cells, that all of these innate-like cells actually have a capacity to remember pathogens or remember kind of insults and respond differently upon a secondary insult. And we've been kind of asking that question within the context of malaria. Does malaria actually shape the innate immune compartment, similar to how what's been described, for example, with vaccine adjuvants, or with BCG, or with kind of this concept of trained immunity? And so when I was a postdoc in Maggie Feeney's lab at UCSF, we were particularly interested in gamma delta T cells and trying to understand how repeated malaria infection actually drove a dysregulated inflammatory response from gamma delta T cells. And more recently, so in my lab at Stanford, we studied how malaria actually might be shaping the natural killer cell response. And so we took advantage. We do these longitudinal cohort studies where we actually follow kids over many years and many infections. And within that setting, we can actually look and see whether or not, for example, the various cells in the innate immune compartment might change over time. And in that paper, I think that you're referencing, what we described was this was a phenomenon where we saw that children who were repeatedly exposed to malaria had expansion of a really unusual NK cell population. These were NK cells, human NK cells that lacked expression of CD56. So they're kind of these CD56 negative NK cells. They have high expression of CD16. They don't respond well to myeloid cytokine stimulation. For example, like if these cells are stimulated with IL-12, IL-15, IL-18, they don't respond well to that stimulation, but they are very effective at performing antibody-dependent cellular cytotoxicity. And what was really interesting about this study is we saw this with repeated exposure. We saw this kind of maturation or adaptation of the NK cell response, and we saw that these NK cells were highly functional. But in our setting, we took advantage of this natural experiment that occurred where the Uganda Ministry of Health came in and sprayed homes with insecticides to drive down malaria transmission. And so we actually followed children before spraying of insecticides and after spraying of insecticides to see how stable this phenotype was. And within 6 months of spraying of insecticides and driving down of transmission, we saw significant declines in the capacity of NK cells to perform ADCC, as well as declines in this particular subset of NK cells, these CD56 negative NK cells. And after 3 years of spraying, the children that lived in this malaria endemic setting, they looked like kids that lived in the malaria non-endemic setting. They had none of these cells that were CD56 negative, for example, really suggesting that malaria might be driving this adaptation of natural killer cells to make them, and a more highly functional population emerges, but it requires repeated exposure to the parasite.
That's really interesting. This effects of repeat exposure and like a permanent. And not, I guess, not a permanent adaptation, right? Because if it reverts, but like a new shift in homeostasis. I think you mentioned something really important I wanted to get back to, which is you've been doing a lot of collaboration. Obviously, you're not going to study malaria patients in the U.S., and so you've developed these collaborations with other governments and groups. Can you talk a little bit about that and how you're successful at doing that? Because I think that's an important piece of information for people interested in this type of work.
I think super important and really kind of really critical. So I'm part of so we work, our Ugandan collaborators with whom we work are an NGO. They're called the Infectious Disease Research Collaboration. I think they're one of the largest nonprofit employers in Uganda. They have several hundred employees. And they're focused on infectious disease. They do a lot of work in malaria, but they do work in HIV. They do work in tuberculosis as well. And we've, so I've been part of this collaboration since I was a trainee at UCSF, but the collaboration actually, has been around for more than 20 years. It was started in the late 1990s. It was a collaboration between Phil Rosenthal and Moses Kamya. And I think the important lesson there is, that these collaborations or this type of work takes time to build. It's difficult to kind of just drop in and say, oh, let me get some samples to kind of study. These are much more, I think, involved in long-term types of collaborations that really should be driven by the questions that are important to our colleagues that are dealing with this disease. And so when the collaboration started in the late 1990s, early 2000s, the main focus of interest was trying to test novel antimalarials or different antimalarials, because at the time chloroquine was the drug that was being used, and it was a suboptimal therapy. And the real need was to do new clinical trials to test alternative antimalarials. That was followed by studies of drug resistance, for example, and the malaria collaboration with IDRC was focused on testing new antimalarials as well as evaluating for drug resistance. Over time, that's evolved, and now the collaboration studies host immunity, for example, studies preventative interventions like chemoprevention. The collaboration studies modifications to houses, for example, to make them less amenable to the Anopheles mosquito habitats, for example. There's just a wide spectrum of things that are done, and I think what's made the collaboration successful is that it's really been driven by the interest and the desires of the team in Uganda who deal with this pathogen on a daily basis.
I come originally from South America, so I think it's so valuable to give countries that are less have less advantages when it comes to biomedical research, the chance to contribute to the research of the diseases that affect their own people. And I think it's so important also in the context of these researchers in these countries, that many of them, I assume, want to stay and work in their own countries. They don't necessarily have to leave in order to have a scientific career. So I think it's so important to keep this kind of collaborations and to really help or to really support research done in kind of the global south or outside of the usual places. So that's very valuable.
Yeah, absolutely. And I think that, we still have a lot of work to do in that space, and think in terms of kind of supporting careers and supporting career development. I think, in North America, Europe, there's Australia, there's a pretty clear kind of route for education, for advancement, for when you can go from being a trainee to being a faculty member to writing successfully for grants. It's that those transitions are much more challenging, and in places like Uganda, where the infrastructure doesn't really exist to truly support that. And I think we as researchers still have a lot of work to do to try to bring those opportunities and real equity in research funding and in research advancement to those settings.
Well, that's a very nice, I think, topic in which to kind of reach the end of our conversation. And I would like to ask you, so we like to ask our guests a little question to get to know a little bit better, to our listeners can have more something besides their research. I think we discussed a lot about your career journey. So I think we did learn a lot about you, but we're going to ask you a different question. If there was one hobby that you always wanted to pursue but never have been able to, what is it?
It's a great question. I think if there's one hobby that I've always wanted to pursue, that I've dabbled, but really not really get into it, is actually being a DJ. I really enjoy DJing. When I was actually at the NIH in the cloister program, we would have these house parties, and I would love DJing those parties. And so something that the feeling of actually playing music and kind of being able to kind of control a crowd it's very exhilarating. Actually, I'd love to maybe be a DJ. My kids though, they're abhorred. They're like, you can't be a DJ.
Oh, kids. Well, I guess you're attending any conference in the future. I think here the organizers, they don't need to get a DJ. You can just ask you.
There you go. They'll be stuck with 90s hip hop though. I don't know. At a certain point, that becomes oldies but goodies. But yeah, that's.
I mean, I would assume that most of us millennials or older would be fine with that. So the younger people, well, they need to adapt.
Yeah. Well, thank you very much for coming on.
Yeah, thanks for having me. And just a plug. April 25th is World Malaria Day, and so I think this will be after that. But it's just a reminder for us to think about malaria and how bad it is, but then also how much. There's a lot of optimism, I think, to kind of over the next period of time that we'll be able to really make a dent in malaria around the world.
Absolutely. Maybe it's also a good moment to ask you if there's any open positions, or if you're looking for researchers to join your lab. Is there anything you want to publicize? This is the time as well.
Yeah, absolutely. Always looking for excited postdocs and students who are interested. So definitely feel free to reach out to us. Our lab website or my email are the best ways to kind of get in touch with us, or also by Twitter. I think our last two postdocs, at least one of them, found us on Twitter. So that's definitely a way to get in touch.
Well, listeners, now you know, if you want some very exciting and very impactful research options, then you know who to contact. Thank you so much for joining us. It's been such a pleasure talking to you.
Yeah, thanks, Brenda. Thanks, Jason.
Thank you. That brings us to the end of our show. Don't forget to subscribe to our newsletter at www.immunologypodcast.com to get the show notes, including an episode summary and links to all the interview and Roundup papers. You can also reach out to us on Twitter at @ImmunoPodcast or via email at info@immunologypodcast.com with feedback or to suggest guests. See you next time.